So hello everyone again and welcome to my channel. In here we'll be looking at the property of enzymes, and it's quite interesting because I've posted videos relating to the properties of enzymes, and in here we'll be continuing from these particular videos. Now one of these is the regulated activity. And what does this imply? This implies that enzymes are susceptible to regulation, and this susceptibility to this particular regulation actually focuses on the catalytic ability or activities that can either increase or decrease the rate or the process of our metabolic needs of our body or our cells. Now, one of these particular steps is the stage called post-translational modification. And what does this imply? This implies a particular stage after you made your mRNA, where the ribosome comes in and actually reads your mRNA, and each codon actually introduces various amino acids that form peptide bonds, and all these new peptide bonds being formed leads to the formation sequence or series of amino acids that then transforms into our protein. Now this protein needs to be able to go from the ribosome or from this particular region over here to a target place, and during this journey or before this journey takes off, what happens is that this particular protein or enzyme actually is being characterized by various functional groups in order for we to be able to have a long-lasting enzyme as it moves from one particular location to another location in order for it to execute its biochemical synthesis or reactions over there. And it's quite crucial as well because this is also important for identifying various ligands or various substrates that is required for the enzyme to function as well properly. So that's about it for the post-translational modification of our particular enzyme or protein over there. Now, next one is the process of allostery. And allostery is quite an interesting topic because in here we're looking at a site other than the active site of a particular enzyme. Now, if you recall from the previous video where we look at a particular enzyme and how this enzyme has a particular region called the active site. Now, this active site only knows a particular substrate or series of substrates, and this series of substrate binds to it and a particular location in this particular site called the catalytic residue, which are or is a particular amino acid that actually causes the reaction of these substrates to form the product is actually present over here, and that is how particular enzymes actually function to transform substrates to our product. However, there might be a couple of regions other than the active site which actually does something different by modifying this particular protein or enzyme over here. This particular region is called the allosteric site. And what does this allosteric site? This allosteric site gives way for a particular molecule to bind to it, and this molecule is called the effector. And what the effector does is that the effector kind of changes after binding the overall structure or the 3D structure of this enzyme to either enhance its activity or inhibit its activity. So that is how this kind of occurs, and this is how this regulatory process enhances the formation or the supporting of metabolic activities in this cell, or limits the metabolic activities of that same cell through a particular reaction process or series of reaction processes. Now, the last one over here is looking at proteolytic cleavage, and this proteolytic cleavage looks at a particular protein or enzyme called the protease, and the protease what it does is that it cleaves various peptide bonds in a protein to form either the polypeptide or it forms amino acids. So that is the purpose of this particular process. This is important as well because there are some proteins that have actually stayed so long, and now if you are making new proteins or new amino acids, you need these proteins to destroy the old ones to give way for the new ones to function fresh and nicely in various chemical processes. Now, our last property of enzyme is called the reversibility. This is an interesting concept because enzymes can catalyze reactions in both ways, which is going in the forward direction or in the reverse direction. And this particular both directional processes actually depends on various conditions, like for example the availability or the amount of our substrate that is present in the system, or we have probably energy that influences this particular process. And energy, for example, can be the bond energy or that you want to cleave or you want to form. All those kind of availability leads to either the reverse direction be favored or the forward direction be favored. And last and foremost, diverse conditions that could actually enhance the regulation of this particular enzyme. Either you have pH involved or you have various ions or salts present in the system that could enhance the activity of this particular enzyme in a particular biochemical reaction. So that's about it for this particular topic. Thanks for the journey of following me through this. I really appreciate it. Please, if you like it, please give it a big thumbs up, subscribe and share, and don't forget to leave a comment and give me a particular question that you have problems with, and I'll be glad to answer everything for you. All the same, guys, have a good day. Peace, love you all, and see ya on my next video. Bye.